Hello, and welcome back to our channel where we dive deep into the wonders of science. It's always a pleasure to have you here with us as we explore the fascinating world of scientific research. Our mission is to bring you the latest discoveries and breakthroughs from the scientific community. Today, I invite you to join me in delving into an exciting and innovative study that promises to capture your interest. We'll be looking at groundbreaking experiments, discussing their implications, and understanding how they can impact our future. So stay tuned and let's embark on this scientific journey together. The research we're discussing today is titled Garlic Straw-Derived Composite Hydrogel via Electron Beam Irradiation for Adsorptive Removal of Methylene Blue. This intriguing study delves into the innovative use of agricultural waste, specifically garlic straw, to address a pressing environmental issue. This study presents a unique approach to environmental sustainability by utilizing garlic straw in the creation of a hydrogel composite. The researchers have employed electron beam irradiation to enhance the properties of the hydrogel, making it highly effective in absorbing pollutants, for the efficient removal of methylene blue from water. Methylene blue is a common dye pollutant, and its removal is crucial for maintaining clean water sources. This research not only offers a solution to water pollution, but also promotes the recycling of agricultural byproducts, contributing to a more sustainable future. Leading this remarkable research is Professor Dr. Ghada Adel from the Polymer Chemistry Department at the National Center for Radiation Research and Technology, Egyptian Atomic Energy Authority, based in Cairo, Egypt. Dr. Ghada Adel has been a pioneer in the field of polymer chemistry for over 2 decades, contributing significantly to advancements in radiation technology. Her dedication and innovative approach have earned her numerous accolades and recognition in the scientific community. Joining her are 2 brilliant scientists, Dr. Mai Elshahawy, who also serves as the corresponding author. Dr. Elshahawy's expertise lies in the synthesis and characterization of novel polymeric materials. Her meticulous research and attention to detail have been instrumental in pushing the boundaries of what is possible in this field. And Dr. Nehad Ahmed, both from the same esteemed department and research center. Dr. Ahmed specializes in the application of radiation technology to develop new materials with unique properties. Her innovative experiments and collaborative spirit have made her a key player in this groundbreaking research team. Together, these 3 scientists are making significant strides in the world of polymer chemistry, paving the way for future innovations. Transition Publication Information. This groundbreaking work has been published in Volume 321 on the 15th of July, 2024, under Article number 129,487. Transition Invitation to the audience. So, get comfortable and join us as we explore the highlights of this innovative research. From the preparation of the polyvinyl alcohol/pectin/garlic straw composite hydrogel to the application of electron beam radiation as a green cross-linking technique, there's much to uncover. Thank you for tuning in today. Don't forget to like, share, and subscribe to stay updated with the latest scientific breakthroughs. Let's dive right into the details of this study. Our modern world thrives on vibrant colors. We see it in our clothes, our homes, and countless industrial products. But this reliance on synthetic dyes has a dark side. Millions of tons of these dyes end up polluting our precious water resources each year. These pollutants don't just taint our water, they poison our ecosystems. Fish struggle to survive, plants wither and die. Even humans face serious health risks from exposure to these toxic chemicals. The situation is dire. We urgently need innovative solutions to combat this growing threat to our planet and our health. In the world of cutting-edge science, sometimes the most unexpected solutions emerge from humble beginnings. A team of researchers led by Professor Dr. Ghada Adel, Dr. Mai Elshahawy, and Dr. Nehad Ahmed turned their attention to a surprising source, garlic straw. This agricultural byproduct, often discarded as waste, holds remarkable potential. The team envisioned transforming this waste into a powerful tool for water purification. Their goal was to create a biodegradable hydrogel from garlic straw that could effectively remove harmful dyes from wastewater. The process of creating this innovative hydrogel is a testament to human ingenuity. The researchers began by extracting cellulose from the garlic straw. This cellulose served as the backbone for their hydrogel. To enhance its properties, they combined it with polyvinyl alcohol, a readily available polymer known for its gel-forming abilities. To further strengthen the gel's structure and improve its dye-absorbing capabilities, they added pectin, a natural gelling agent found in fruits. The final ingredient in this chemical cocktail was electron beam irradiation. This process helped to cross-link the polymers, forming a stable and porous hydrogel network. The true test of any water purification technology lies in its ability to remove contaminants effectively. The team chose methylene blue as a model dye for their study. Methylene blue is commonly used in various industries, but poses significant environmental risks. They introduced the garlic straw hydrogel into water contaminated with methylene blue. The results were remarkable. The hydrogel exhibited an impressive capacity to absorb the methylene blue dye, effectively removing it from the water. This successful demonstration proved the potential of their creation as a viable solution for dye-contaminated wastewater treatment. To optimize the hydrogel's performance, the researchers delved deeper into the mechanisms behind its dye removal capabilities. They investigated the adsorption process in detail, studying the interaction between the hydrogel and the methylene blue molecules. Their findings revealed that the adsorption process primarily occurred through electrostatic interactions. The positively charged methylene blue molecules were attracted to the negatively charged functional groups present on the hydrogel's surface. This attraction led to the dye molecules being trapped within the porous structure of the hydrogel, effectively removing them from the water. The researchers didn't stop at simply demonstrating the hydrogel's effectiveness. They wanted to understand how its performance varied under different conditions. They conducted a thermodynamic analysis to determine the effective temperature on the adsorption process. Their findings revealed that the adsorption of methylene blue onto the garlic straw hydrogel was an endothermic process. This means that the adsorption process was favored at higher temperatures. This insight is crucial for optimizing the hydrogel's performance in real-world applications, where temperature fluctuations are inevitable. The successful development of this garlic straw hydrogel is a groundbreaking achievement in the field of environmental science. It holds immense promise for addressing the pressing issue of dye pollution in our water bodies, which has been a persistent problem for decades. Its ability to effectively remove methylene blue, a model dye pollutant, demonstrates its efficiency and potential for broader applications. This highlights its potential for treating wastewater from various industries. Textile factories, in particular, known for their heavy reliance on dyes, produce large volumes of colored effluents that are challenging to treat. These factories could significantly benefit from integrating this innovative technology into their effluent treatment processes, thereby improving their environmental footprint. By removing harmful dyes before they are discharged into natural water bodies, we can protect our rivers, lakes, and groundwater from contamination, ensuring that these vital resources remain unpolluted. This will safeguard aquatic life, which is often severely affected by dye pollutants, maintain the ecological balance of our water bodies, and support the biodiversity that thrives in these environments. Additionally, ensuring the availability of safe and clean water for human consumption is crucial for public health. This technology not only addresses environmental concerns, but also contributes to the well-being of communities by providing access to uncontaminated water sources. Beyond methylene blue, expanding the scope. While the initial focus of the research was on removing methylene blue, the potential applications of this garlic straw hydrogel extend far beyond a single dye. Its unique properties and efficient adsorption capabilities make it a promising candidate for tackling a broader range of water pollutants. Further research is underway to explore its effectiveness in removing other common dyes, heavy metals, and even pharmaceutical residues from wastewater. This versatility positions the garlic straw hydrogel as a potential game-changer in the fight against water pollution, offering a sustainable and cost-effective solution for a cleaner and healthier future. A global challenge, a local solution, the power of innovation. Water pollution is a pressing issue that affects millions of people worldwide, contaminating vital water sources and posing serious health risks. The development of this innovative hydrogel exemplifies the power of scientific research in addressing global challenges through local solutions. This hydrogel, designed to purify water, is a testament to the ingenuity and dedication of scientists who strive to make a difference. By transforming garlic straw, an abundant agricultural byproduct often discarded as waste, into a valuable resource for water purification, researchers have found a way to turn waste into a solution. This not only helps in managing agricultural waste but also provides a sustainable method for cleaning polluted water. The research team has demonstrated a commitment to sustainability and circular economy principles. Their work highlights the importance of rethinking how we use and reuse resources, ensuring that nothing goes to waste. This approach not only tackles the problem of dye pollution, but also promotes resource efficiency and waste reduction. By using innovative methods to clean water, we can significantly reduce the environmental impact of industrial processes. Such innovative solutions are crucial in our collective efforts to protect our environment and ensure a sustainable future for generations to come. Community involvement and awareness are key to driving these changes and making a lasting impact. By investing in research and fostering collaboration between scientists, policymakers, and industries, we can create a unified approach to solving environmental issues. This collaboration ensures that scientific advancements are supported by effective policies and practical applications. We can pave the way for a cleaner and healthier planet. Together, through innovation, collaboration, and dedication, we can overcome the challenges of water pollution and create a sustainable future for all. Champions of Change, the research team. The groundbreaking work of Professor Dr. Ghada Adel, Dr. Mai Elshahawy, and Dr. Nehad Ahmed deserves recognition and applause. Their dedication to finding sustainable solutions for environmental challenges, coupled with their innovative spirit, has resulted in a potentially game-changing technology. Their efforts serve as an inspiration to aspiring scientists and a testament to the power of collaborative research in tackling real-world problems. Their work reminds us that even amidst global challenges, ingenuity and determination can pave the way for a brighter future. A cleaner future. The promise of sustainable solutions. In a world where environmental challenges are becoming increasingly complex, the need for innovative and sustainable solutions has never been more urgent. As we strive towards a future where clean water is accessible to all, innovations like the garlic straw hydrogel offer a beacon of hope. This remarkable material, derived from natural sources, has the potential to revolutionize water purification processes, making them more efficient and environmentally friendly. This groundbreaking research signifies a crucial step towards achieving sustainable solutions for water purification. The dedication and hard work of scientists and researchers are paving the way for a future where clean water is not just a dream, but a reality for everyone. By harnessing the power of nature and combining it with human ingenuity, we can effectively address the growing threat of water pollution and protect our planet's most precious resource. The integration of natural elements with cutting-edge technology is key to developing sustainable methods that can be implemented on a global scale. The journey towards a cleaner future requires collective action and a commitment to innovation. Communities, scientists, and policymakers must work together to create and implement strategies that will ensure the availability of clean water for generations to come. By supporting research, promoting sustainable practices, and embracing a shared responsibility for our planet, we can create a world where clean water is not a privilege, but a right enjoyed by all. Together, we can make a significant impact and leave a lasting legacy of sustainability for future generations. Water, the lifeblood of our planet, essential for all living things. Yet this precious resource is under threat. Pollution casts a shadow over our waters. Industrial waste, agricultural runoff, and untreated sewage contaminate our rivers, lakes, and oceans. This pollution poses a grave danger to human health and the delicate balance of our ecosystems. Among the pollutants lurking in our water, synthetic dyes stand out as a significant concern. These dyes are not just a visual blight, they pose serious environmental and health risks. These dyes, widely used in industries like textile and paper manufacturing, often end up in our waterways. The discharge from these industries can turn clear streams into toxic colored channels. One such dye, methylene blue, demands our attention. Known for its vivid blue color, it is used in various applications, from scientific research to medical treatments. A common sight in laboratories and medical treatments, methylene blue, in high concentrations, can be harmful to aquatic life and humans alike. Its widespread use makes it a persistent pollutant. It can disrupt oxygen levels in water, suffocating fish and other organisms. The dye's presence in aquatic environments can lead to devastating ecological consequences. In humans, exposure to high levels of methylene blue can cause a range of health problems, from nausea and vomiting to more serious complications. Long-term exposure can lead to chronic health issues. The presence of methylene blue in our water sources is a stark reminder of the impact human activities have on the environment. It highlights the urgent need for better waste management practices. Finding effective ways to remove this and other harmful dyes from our water is not just an environmental challenge, it's a matter of global health and well-being. Innovative solutions and community efforts are essential in tackling this issue. As we search for solutions to combat water pollution, nature often offers a helping hand. Scientists and researchers are increasingly turning to sustainable and eco-friendly methods for water purification. One such innovation comes from an unexpected source: garlic. Not the cloves themselves, but the often discarded straw. Garlic straw, a byproduct of the garlic industry, has shown remarkable potential in the fight against water pollution. Researchers have discovered that garlic straw can be transformed into a powerful tool for removing pollutants like methylene blue from water. This transformation involves converting the garlic straw into a composite hydrogel, a substance with a remarkable ability to absorb and trap certain pollutants. The process of creating this innovative hydrogel begins with collecting and cleaning garlic straw. The straw is then treated to break down its structure and extract valuable components. These components, rich in cellulose and other natural polymers, form the building blocks of the hydrogel. Through a carefully controlled chemical process, these building blocks are cross-linked, creating a three-dimensional network capable of holding large amounts of water. This network acts like a sponge, but with a crucial difference. It's designed to selectively trap specific pollutants like methylene blue while allowing water to pass through. The resulting garlic straw composite hydrogel is a testament to the ingenuity of scientists and the power of nature-inspired solutions. It's a sustainable and eco-friendly alternative to conventional water treatment methods that often rely on harsh chemicals. Section 5. The Dance of Removal: How the Hydrogel Captures Methylene Blue. The magic of the garlic straw composite hydrogel lies in its adsorption process, a fascinating mechanism that sets it apart from other materials. Adsorption, unlike absorption, is a surface phenomenon where molecules adhere to the surface rather than being absorbed into the material. Imagine the hydrogel as a vast dance floor, and the methylene blue molecules as eager dancers ready to join the party. The surface of the hydrogel, with its specific chemical properties, acts like an irresistible invitation to the methylene blue molecules, drawing them in. Drawn by forces of attraction, the methylene blue molecules move toward the hydrogel's surface and latch onto specific binding sites, much like dancers finding their perfect partners. These binding sites, like perfect dance partners, hold onto the methylene blue molecules tightly, preventing them from escaping back into the water, ensuring they stay put. As more and more methylene blue molecules are drawn to the hydrogel's surface, the water gradually loses its blue tint, becoming clearer and cleaner, a visible transformation. This elegant dance between the hydrogel and the methylene blue molecules effectively removes the dye from the water, making it safe for consumption and protecting aquatic life, showcasing the hydrogel's remarkable efficiency. Section 6. A Promising Dawn: Research Findings and the Effectiveness of Garlic Straw Hydrogel. The development of the garlic straw composite hydrogel has generated immense excitement in the scientific community. Rigorous laboratory tests have confirmed its effectiveness in removing methylene blue from water. These tests have revealed that the hydrogel can remove a significant percentage of methylene blue from contaminated water in a relatively short amount of time. Furthermore, the hydrogel has demonstrated impressive reusability. After capturing the methylene blue, the hydrogel can be treated to release the dye, allowing it to be used multiple times. This reusability makes the garlic straw composite hydrogel a cost-effective and sustainable solution for water treatment. The promising results of these studies have paved the way for further research and development, bringing hope for a cleaner and healthier future. Section 7. beyond the laboratory, real-world applications, and future directions. While still in its early stages, the garlic straw composite hydrogel holds immense potential for real-world applications. This innovative material is being closely studied for its unique properties and versatility. Its ability to effectively remove methylene blue from water makes it a promising candidate for treating industrial wastewater. This could revolutionize how industries manage their waste, ensuring cleaner water discharge. Textile factories in particular could benefit from this innovation, reducing their environmental impact by treating their dye-laden wastewater before it's released into the environment. This not only helps the environment, but also aligns with stricter regulations. Beyond industrial applications, the hydrogel could also play a significant role in purifying drinking water, especially in developing countries where access to clean water is limited. This could drastically improve public health. Its low cost and ease of use make it an ideal solution for point-of-use water treatment, providing safe drinking water to communities in need. This could be a game-changer for many rural areas. The future of the garlic straw composite hydrogel is bright, with ongoing research exploring its potential for removing other pollutants, such as heavy metals and pesticides. This could open new avenues for environmental protection and sustainability. The development of the garlic straw composite hydrogel is a shining example of how innovation can pave the way for a more sustainable future. By utilizing a waste product like garlic straw, this technology promotes a circular economy, reducing waste and maximizing resource utilization. Its environmentally friendly nature extends beyond its raw materials. The hydrogel's ability to remove pollutants without the need for harsh chemicals minimizes the risk of secondary pollution, protecting both human health and the environment. By embracing such sustainable solutions, we can move towards a future where clean water is not a privilege, but a right for all. The success of the garlic straw composite hydrogel highlights the importance of investing in research and development, particularly in the field of sustainable technologies. Governments, industries, and research institutions must work together to foster innovation and bring such groundbreaking solutions to the forefront. But innovation alone is not enough. We, as individuals, must also play our part. By making conscious choices in our daily lives, from reducing water consumption to supporting businesses that prioritize sustainability, we can contribute to a cleaner and healthier planet. The fight against water pollution is a shared responsibility. Let us embrace innovation and work together to ensure a future where clean water flows freely for generations to come. Addressing the global water crisis requires a collaborative effort. International cooperation is crucial for sharing knowledge, resources, and best practices. Developed nations have a responsibility to support developing countries in their pursuit of sustainable water management solutions. Collaboration between scientists, engineers, policymakers, and local communities is essential for developing and implementing effective water treatment technologies. By working together, we can overcome the challenges of water pollution and ensure water security for all. The garlic straw composite hydrogel is a testament to the power of human ingenuity when we work together for a common goal. The story of the garlic straw composite hydrogel is a beacon of hope in the face of environmental challenges. It demonstrates that even the most unexpected sources can hold the key to groundbreaking solutions. By embracing innovation, sustainability, and collaboration, we can create a legacy of clean water for generations to come. Let us be inspired by this remarkable innovation and continue to explore new frontiers in water purification. Together, we can ensure that the lifeblood of our planet flows clean and pure, nurturing life and sustaining our world for generations to come. The future of water is in our hands. Let us choose wisely and act decisively to protect this precious resource. Hey there, science fans! Today we're diving into the exciting world of material science. We're talking about stuff that can revolutionize industries from medicine to agriculture. Imagine a world with custom-designed materials that are stronger, lighter, and more sustainable. That's the promise of advanced materials, and electron beam or EB radiation is one of the keys to unlocking their full potential. EB radiation is like a super-powered beam of energy that can change the properties of materials at the molecular level. It's like having a microscopic construction crew that can rearrange atoms and molecules to create entirely new substances. This technology is already used in various fields, from sterilizing medical equipment to modifying the properties of plastics. In this essay, we'll focus on how EB radiation affects a specific type of material called polyvinyl alcohol and pectin hydrogels, or PVA-PT hydrogels. These hydrogels are like super absorbent sponges that can hold up to 1,000 times their weight in water. They have a wide range of applications, including drug delivery, wound healing, and agriculture. Get ready to explore the fascinating world of EB radiation and discover how it's shaping the future of PVA-PT hydrogels. So, what exactly is electron beam radiation? Imagine a stream of tiny particles called electrons traveling at incredibly high speeds. These electrons carry a lot of energy, and when they collide with a material, they can cause some pretty amazing changes. Think of it like hitting a billiard ball with a cue stick. The cue stick transfers energy to the ball, causing it to move. Similarly, EB radiation transfers energy to the molecules in a material, causing them to vibrate, rotate, and even break apart. This energy transfer can lead to a process called crosslinking, where the molecules in a material form new bonds with each other. It's like weaving a fabric together, making it stronger and more durable. In the case of PVA-PT hydrogels, EB radiation can create crosslinks between the polymer chains, improving their mechanical strength, swelling behavior, and thermal stability. EB radiation offers several advantages over other cross-linking methods. It's a fast, clean, and efficient process that doesn't require any harsh chemicals or solvents. This makes it an environmentally friendly option for modifying materials. Now, let's talk about the stars of our show, PVA-PT hydrogels. These innovative materials are revolutionizing various fields with their unique properties and versatility. These amazing materials are made from two main components, polyvinyl alcohol, or PVA, and pectin, or PT. Each of these components brings its own set of remarkable characteristics to the table. PVA is a synthetic polymer known for its excellent film-forming properties, biocompatibility, and water solubility. It's widely used in various industries due to its ability to form strong, flexible films. It's like a long chain of beads, with each bead representing a repeating unit of the polymer. This structure gives PVA its strength and flexibility, making it an ideal component for hydrogels. Pectin, on the other hand, is a natural polysaccharide found in the cell walls of plants. It's a key ingredient in the food industry, especially in the production of jams and jellies. It's what gives fruits like apples and oranges their gel-like texture. This natural gelling agent is not only edible, but also biodegradable and non-toxic, making it environmentally friendly. Pectin is known for its biodegradability, non-toxicity, and ability to form gels in the presence of water. This makes it an excellent choice for creating hydrogels that are safe for both medical and environmental applications. When PVA and PT are combined, they create a unique material with a network-like structure. This structure is what gives the hydrogel its remarkable properties. This network is filled with tiny pores that can hold large amounts of water, just like a sponge. This high water content is crucial for applications that require moisture retention. The properties of PVA-PT hydrogels can be further enhanced by adding other materials, like nanoparticles, drugs, or even natural fibers. These additives can tailor the hydrogel for specific uses, making it even more versatile. These hydrogels have a wide range of potential applications in various fields. Their versatility and adaptability make them suitable for numerous innovative solutions. In medicine, they can be used as wound dressings, drug delivery systems, and even as scaffolds for tissue engineering. Their ability to maintain a moist environment and deliver therapeutic agents makes them invaluable in healthcare. And scaffolds for tissue engineering. These scaffolds can support cell growth and tissue regeneration, offering new possibilities in regenerative medicine. In agriculture, they can be used to improve soil water retention, deliver fertilizers, and protect seeds from harsh environmental conditions. This can lead to more efficient water usage and better crop yields, and protect seeds from harsh environmental conditions. By providing a controlled release of water and nutrients, these hydrogels can help plants thrive even in challenging environments. Section 4. Garlic straw, a natural wonder material. In our quest to develop even more sustainable and eco-friendly materials, researchers have turned to nature for inspiration. One such natural resource is garlic straw, the often overlooked byproduct of garlic harvesting. Garlic straw is typically discarded as agricultural waste, but it turns out to be a treasure trove of valuable components. It's rich in cellulose, a natural polymer that provides strength and structure to plants. Cellulose can be extracted from garlic straw and used to reinforce PVA-PT hydrogels, making them stronger and more durable. But that's not all. Garlic straw also contains lignin, another natural polymer that acts as a binder, and hemicellulose, which improves the water absorption capacity of the hydrogels. By incorporating garlic straw into PVA-PT hydrogels, we can create composite materials that are not only biodegradable and biocompatible, but also possess enhanced properties. This approach aligns with the principles of green chemistry, which encourages the use of renewable resources and minimizes waste generation. By utilizing garlic straw, we can reduce our reliance on synthetic polymers and contribute to a more sustainable future. Section 5. Preparing PVA/PTGS composite hydrogels. Now that we know about the amazing properties of PVA-PT hydrogels and garlic straw, let's see how we can combine them to create even more remarkable materials. The synergy between these components can lead to innovative applications in various fields. The preparation of PVA/PTGS composite hydrogels is a fascinating process that involves several carefully controlled steps. Each step is crucial to ensure the final product has the desired properties. First, we need to extract cellulose from garlic straw. Garlic straw is an abundant agricultural byproduct, making it a sustainable source of cellulose. This can be done using a variety of methods, including chemical treatments and mechanical processes. One common approach is to treat the straw with chemicals that break down the other components, leaving behind pure cellulose. This step is essential to remove impurities and obtain high-quality cellulose. The extracted cellulose is then washed, dried, and purified to ensure it is free from any residual chemicals. After drying, the cellulose is ground into a fine powder. This powder form is easier to mix with other materials. Next, we prepare a solution of PVA and PT in water. Polyvinyl alcohol, PVA, is a synthetic polymer known for its excellent film-forming and adhesive properties, while platinum PT nanoparticles enhance the hydrogel's mechanical strength and conductivity. This solution acts as a matrix for the cellulose to be incorporated into. The uniform distribution of PT nanoparticles within the PVA solution is critical for the composite's performance. The cellulose powder is slowly added to the PVA-P solution while stirring continuously. This step requires precision to ensure the cellulose is evenly distributed. This ensures that the cellulose is evenly dispersed throughout the mixture, preventing clumps from forming. An even dispersion is key to achieving a uniform hydrogel structure. Once the cellulose is fully dispersed, the mixture is poured into molds. The choice of mold shapes can influence the final application of the hydrogel and exposed to EB radiation. Electron beam, EB radiation, is used to initiate the cross-linking process. The EB radiation initiates cross-linking between the PVA, PT, and cellulose molecules, forming a strong and interconnected network. This cross-linking enhances the mechanical properties and stability of the hydrogel, forming a strong and interconnected network. The resulting network structure is crucial for the hydrogel's functionality. The result is a PVA/PGS composite hydrogel with enhanced properties. These hydrogels can be used in a variety of applications, including biomedical devices, environmental remediation, and smart materials. Section 6. Gel content analysis, measuring the strength. So, how do we know if our PVA/PTGS composite hydrogels are as strong as we think they are? This is a crucial question because the strength of these hydrogels determines their suitability for various applications, from medical uses to industrial purposes. That's where gel content analysis comes in. This method is essential for understanding the structural integrity of our hydrogels. This technique allows us to measure the percentage of cross-linked polymer in our hydrogels, which is directly related to their strength and stability. Cross-linking is a process where polymer chains are linked together, providing the material with enhanced mechanical properties. To perform gel content analysis, we first weigh a dry sample of our hydrogel. This initial weight is crucial as it serves as the baseline for our calculations. Then we immerse the sample in a solvent that can dissolve any uncross-linked polymer chains. The choice of solvent is important as it must effectively dissolve the non-cross-linked portions without affecting the cross-linked network. After a certain amount of time, we remove the sample, dry it again, and weigh it. This drying process ensures that we only measure the weight of the cross-linked polymer. The difference in weight between the initial dry sample and the final dry sample represents the amount of uncross-linked polymer that was dissolved in the solvent. This step is critical for determining the gel content accurately. By dividing the weight of the final dry sample by the weight of the initial dry sample and multiplying by 100%, we can calculate the gel content of our hydrogel. This calculation gives us a clear picture of the extent of cross-linking within the material. A higher gel content indicates that a larger percentage of the polymer chains are cross-linked, resulting in a stronger and more stable hydrogel. This is particularly important for applications requiring high mechanical strength and durability. By varying the concentration of garlic straw and the dose of EB radiation, we can fine-tune the gel content of our PVA/PTGS composite hydrogels to meet specific application requirements. This flexibility allows us to customize the hydrogels for a wide range of uses, ensuring optimal performance in each scenario. Hey there, science fans! We're diving into the fascinating world of adsorption and how it can help us clean up our water. Specifically, we're looking at how we can remove a common dye called methylene blue. You might recognize it from those cool tie-dye experiments. Methylene blue is often used in industries like textiles and paper. However, it can end up polluting our precious water sources. That's where our heroes come in, composite hydrogels. These amazing materials act like sponges, soaking up the methylene blue and preventing it from harming our environment. In our science lab, we're studying how different factors affect how well these hydrogels can remove methylene blue from water. It's all about understanding the adsorption kinetics, how fast the dye sticks to the hydrogel. So, put on your lab coats and let's get started. Clean water is essential for all living things, and finding effective ways to remove pollutants is crucial. Adsorption is a powerful technique that uses materials like our composite hydrogels to remove contaminants from water. Imagine it like a game of catch, where the hydrogel is the glove and the methylene blue dye is the baseball. These hydrogels are made of special materials like polyvinyl alcohol platinum, PVA-PT, and PVA/PT garlic straw, GS. Yes, you heard that right, garlic straw. This agricultural waste product can actually be incredibly useful in removing pollutants. These materials are carefully chosen for their ability to attract and hold onto the methylene blue dye, effectively removing it from the water. Our goal is to understand exactly how these materials work and how we can make them even better at cleaning up our water. Now let's talk about adsorption kinetics. This might sound complicated, but it's really about understanding how fast the methylene blue dye sticks to our composite hydrogels. Think of it like solving a puzzle. We need to figure out what factors affect how quickly the puzzle pieces, the dye molecules, fit into their slots, the hydrogel. To do this, we use mathematical models that help us describe this process. These models, with names like pseudo-first order and pseudo-second order, might sound intimidating, but they're just tools that help us understand the adsorption process better. By studying these models and comparing them to our experimental data, we can determine which model best describes how the methylene blue interacts with our composite hydrogels. This knowledge is essential for designing and optimizing water treatment processes. Let's start with the pseudo-first order model, a simple way to describe adsorption kinetics. This model assumes that the rate at which the dye molecules stick to the hydrogel depends on how many empty spaces are left on the hydrogel's surface. Imagine it like finding a parking spot. The more empty spots there are, the easier it is to find one quickly. This model uses a few key parameters to describe the adsorption process: Kexp, Qcal, K1, and R2. Kexp is the amount of dye adsorbed at a specific time, while Qcal is the amount predicted by the model. K1 tells us how fast the adsorption happens, and R2 tells us how well the model fits our experimental data. The closer R2 is to 1, the better the fit. Section 5, Pseudo-Second Order Model, a better fit for our data. While the Pseudo-First Order Model is a good starting point, we found that the Pseudo-Second Order Model actually fits our experimental data much better. This model suggests that the rate of adsorption depends on both the number of empty sites on the hydrogel and the concentration of the dye in the water. Think of it like this. The more dye molecules there are in the water, the more chances they have to bump into and stick to the hydrogel. This model is particularly useful for describing chemisorption, where the dye molecules form strong chemical bonds with the hydrogel surface. Chemisorption is like making a strong friendship. Once the bond is formed, it's less likely to break. The pseudo-second-order model helps us understand this stronger interaction between the methylene blue and the composite hydrogels. Section 6. The Elovich model, when things get a bit more complex. Now, let's explore the Elovich model, which comes in handy when the adsorption process gets a bit more complicated. This model considers that the hydrogel's surface is not perfectly uniform, and that some sites might be more favorable for adsorption than others. Imagine it like finding the perfect spot on a crowded beach. Some spots might be more desirable than others, and they'll fill up first. The Elovich model is particularly useful for describing chemisorption processes, where the number of active sites on the hydrogel decreases over time. As more dye molecules stick to the hydrogel, it becomes harder for others to find those prime adsorption spots. This model helps us understand the dynamics of adsorption when dealing with more complex hydrogel surfaces. Section 7. Intraparticle diffusion model, a journey into the material. Hold on tight, because we're about to journey into the fascinating world of intraparticle diffusion. This model helps us understand how the methylene blue dye molecules move within the pores of the composite hydrogel. Think of it like exploring a maze. The dye molecules have to navigate the twists and turns within the hydrogel to reach the adsorption sites. The intraparticle diffusion model helps us determine if this movement within the hydrogel is a limiting factor in the overall adsorption process. By analyzing the data, we can gain valuable insights into the hydrogel's structure and its impact on the adsorption process. This knowledge is crucial for designing efficient and effective hydrogels for water treatment. Section 8. Temperature's effect on adsorption. Turning up the heat. Now, let's turn up the heat and see what happens. Temperature plays a crucial role in adsorption kinetics. Generally, increasing the temperature speeds up the adsorption process. Think of it like adding more energy to a game of tag. The higher the temperature, the faster the dye molecules move, increasing their chances of colliding with and sticking to the hydrogel. However, it's important to find the sweet spot. If the temperature is too high, it can actually decrease the adsorption capacity. Imagine it like melting ice cream. If it gets too warm, it loses its structure and can no longer hold on to the toppings. Finding the optimal temperature is key for maximizing the efficiency of the adsorption process. Section 9. PVA-PT and PVA/PTGS, the dynamic duo of adsorption. In our quest to find the best materials for removing methylene blue, we've been experimenting with two promising candidates, PVA-PT and PVA/PTGS. Both materials have shown excellent adsorption capabilities, but they have some key differences. PVA-PT is like a reliable workhorse. It consistently removes methylene blue from water. However, PVA/PTGS, with the addition of garlic straw, takes it to the next level. This eco-friendly addition enhances the hydrogel structure, creating more surface area for the dye molecules to stick to. It's like adding more parking spots to our parking lot. Our results show that PVA/PTGS consistently outperforms PVA-PT in removing methylene blue, making it a promising candidate for real-world water treatment applications. Section 10. Rate constants and thermodynamic parameters. Unraveling the secrets. To fully understand the adsorption process, we need to delve into the world of rate constants and thermodynamic parameters. Rate constants, like the ones we discussed earlier, K1 and K2, tell us how fast the adsorption happens. Thermodynamic parameters, on the other hand, provide valuable insights into the energy changes associated with the adsorption process. For example, the Gibbs free energy, dG, tells us if the adsorption is spontaneous or not. A negative dG means that the process happens naturally, without the need for any external force. By analyzing these parameters, we can gain a deeper understanding of the forces driving the adsorption process. Section 11. Conclusion: a brighter future for clean water. As we conclude our exploration of methylene blue adsorption on composite hydrogels, one thing is clear: these amazing materials hold immense potential for cleaning up our water. By understanding the adsorption kinetics, the roles of different kinetic models, and the influence of factors like temperature and material composition, we can design and optimize highly efficient water treatment processes. Our research on PVA-PT and PVA/PTGS highlights the exciting possibilities of using composite materials, especially those incorporating agricultural waste products like garlic straw for environmental remediation. Together, with the power of science and innovation, we can create a brighter and cleaner future for generations to come. Stay curious, my friends.
As we come to the end of our discussion today, we would like to extend our deepest gratitude to the brilliant minds behind this remarkable research. Their dedication and hard work have paved the way for groundbreaking discoveries that will benefit society for years to come. A special thanks to Professor Dr. Ghada Adel, the lead researcher, whose vision and leadership have been instrumental in guiding this project to success. Along with Dr. Mai Elshahawy, the corresponding author, whose meticulous attention to detail ensured the accuracy and reliability of the findings, and Dr. Nehad Ahmed, whose innovative approaches and tireless efforts have been invaluable. All three of these talented scientists are from the Polymer Chemistry Department at the National Center for Radiation Research and Technology. Their expertise in polymer chemistry has opened new avenues for research and development. This esteemed institution is part of the Egyptian Atomic Energy Authority in Cairo, Egypt. The collaborative environment and state-of-the-art facilities at this center have provided the perfect backdrop for their pioneering work. We also want to acknowledge the support staff and fellow researchers who have contributed to this project. Their behind-the-scenes efforts have been crucial in bringing this research to fruition. It is through the collective efforts of these dedicated individuals that we are able to make strides in scientific discovery and innovation. We look forward to seeing the future advancements that will stem from this incredible work. Thank you once again to everyone involved. Your contributions are truly appreciated and will have a lasting impact on the scientific community and beyond. We truly appreciate the exceptional effort and dedication that went into this study. The countless hours spent in the lab, the meticulous attention to detail, and the unwavering commitment to advancing scientific knowledge have not gone unnoticed. Your hard work is the backbone of this groundbreaking research. Your innovative approach to utilizing garlic straw and electron beam irradiation for environmental sustainability is nothing short of inspiring. By thinking outside the box and pushing the boundaries of conventional methods, you have opened new avenues for sustainable practices. This pioneering work not only addresses current environmental challenges, but also sets a precedent for future research. Thank you for contributing such valuable research to the scientific community. Your findings will undoubtedly serve as a cornerstone for future studies and inspire other researchers to explore new possibilities. The impact of your work extends beyond the laboratory, influencing policies and practices that benefit society as a whole. And to our wonderful viewers here on this incredible journey of scientific discovery, we want to extend our heartfelt gratitude. Thank you for joining us today and for being such an integral part of our community. Your continued support and enthusiasm for science mean the world to us. It's your curiosity and passion that drive us to explore new frontiers. We look forward to bringing you more groundbreaking research, insightful discussions, and exciting discoveries in our upcoming episodes. Stay tuned and let's continue this amazing journey together. Thank you once again for your unwavering support. Stay tuned for more exciting content, and don't forget to like, share, and subscribe so you never miss an update. Until next time, take care and keep exploring the wonders of science.